Assalamualaikum and good day everyone, and greetings to our lecturer Muhammad Ishari bin Abdul Halim. We are from class 10, group F, representing the Faculty of Applied Science here at UiTM. Today we are excited to present our findings for experiment 5, gas chromatography, or more commonly known as GC. This experiment is a core part of our CHM260 Basic Instrumental Analysis course, where we explore the powerful technique used to separate and analyze volatile compounds. Hi, my name is Khadija, and this is my group members. We have Irfan and Intan.
First and foremost, we need to know what is gas chromatography is. So gas chromatography is a technique for separating components based on their volatilities based on difference of boiling point. In gas chromatography, the components of a vaporized sample are separated as a result of being partitioned between a mobile gaseous phase and a liquid or a solid stationary phase held in the column. So gas is called carrier gas, and mobile phase are generally inert gases such as helium, argon or nitrogen. So the mobile phase is a liquid or a gas that carries a sample through a solid stationary phase. As a sample, in the mobile phase passes through the stationary phase, the compound in the mixture will separate because of the difference in their affinities for the stationary phase and the difference in their solubilities in the mobile phase. Therefore, there are two types of gas chromatography that we need to know, which is first, gas liquid chromatography GLC, which basically mobile phase is a gas and stationary phase is a liquid. Next, we have gas solid chromatography GSC, which mobile phase is a gas and stationary phase is a solid. The objective of this experiment is firstly to determine the retention time of N-butanol and true-propanol. Secondly, to identify the components present in a standard mixture based on the retention time. Thirdly, to identify the component present in the unknown sample. And lastly, to determine the effect of temperature on retention time and resolution. For this experiment, we use dropper, syringe and beaker for the apparatus, and we use N-butanol, true-propanol, unknown sample and standard mixture of true-propanol and N-butanol with a ratio of 1:1 for chemical.
Moving into the methodology, let's look at how a gas chromatography system is actually built. On this slide, we have two diagrams, which is figure 1 and figure 2, which represent the internal anatomy in the GC instrument. You can see the process begin on the far left with a gas cylinder, which provides the push for our experiment. As we trace the line, the sample moves through the inlet into the coil room located inside the oven and finally reach the detector. Essentially, the GC acts like a high-tech filter. We put a mixture in one end and it uses gas to push that mixture through a very long thin tube to separate the different parts before they reach the computer for analysis. To understand how that separation happens, we need to look at the 6 components of the instrument. Firstly, we have carrier gas. This is our mobile phase. It is usually an inert gas like helium or nitrogen that carries our sample through the system without reacting with it. The second is sample injection part. This is the entry of the point. It is kept hot so that when we inject a liquid sample, it instantly vaporizes into a gas. The third one is chromatography column. This is the heart of the machine. It is the stationary phase where actually separation occurs. Number 4 is column oven. Separation is sensitive to the heat. So this oven controls the temperature to keep the process consistent and efficient. Number 5 is detector. This is the component as they exit the column and converts the present into an electrical signal. And lastly, we have data processing system. Finally, the computer records those signals and turns them into a chromatogram, the graph we use to identify what was in our sample. Okay, now how do we actually perform the experiment? It starts with sample handling. Precision is key here is we rinse the strings to avoid contamination, hold it vertically to remove air bubbles, and ensure we have the exact required volume at eye level before injecting. The basic principle of TC can be summarized in 4 steps. The first one is the liquid sample is injected and vaporized at the inlet. The second one is the carrier gas transport those vapors into the column. The third one is the most important step, which is the components separate based on their boiling points and how they interact with the stationary phase. Imagine a race where some runners get distracted by water station while others run straight through. The ones who don't stop reach the finish line first. Finally, the detector picks up these separated runners and generates the chromatogram that you see on the screen. This allows us to take a complex mixture and identify every single ingredient within it.
These are gas chromatography result for two alcohol samples. The left chromatogram is for n-butanol and the right chromatogram is 2-propanol, and also for standard mixture and unknown. In each graph, the x-axis shows the retention times and the y-axis shows detector response. Each sharp peak represents the compound detected by the GC. The position of the peak shows when the compound comes out of the column, and the area of the peak shows how much of the compound is present. Since each sample shows one major peak, it indicates that the samples are mostly pure and correctly identified. Next is for discussion. Retention time is the time taken. Lastly, to conclude, gas chromatography successfully determined the retention times of 2-propanol and n-butanol. 2-propanol consistently elute before n-butanol, and increasing oven temperature reduced the retention times of both compounds. The experiment demonstrated the usefulness of retention time for compound identification and showed the effect temperature on chromatographic separation. And lastly, to conclude, gas chromatography successfully determined the retention times of 2-propanol and n-butanol. 2-propanol consistently elute before n-butanol, and increasing oven temperature reduced the retention times of both compounds. The experiment demonstrated the usefulness of retention time for compound identification and showed the effect of temperature on chromatographic separation.